Thanks for inviting me to speak here today as well. So I'm just going to talk a little bit about some of the gene editing work that I've done in human induced pluripotent stem cells, which we are using to model neurological diseases. So I should mention I'm funded by the STEM Bank project. STEM Bank is a very, very loose acronym for stem cells for biological assays of novel drugs and predictive toxicology. And so the goal of this project is to take biopsies from about 500 different patients with various neurological conditions, including neurodegenerative diseases such as Parkinson's and Alzheimer's, peripheral neuropathies and neurodysfunctional disorders. And on top of all that, we're also investigating diabetes. And with those the biopsies, we're generating iPS cell lines. And we're also producing very well-defined protocols for differentiating these cells into disease-relevant cell types, which are then being used in drug screening assays and also being used in basic research. So this is a pretty large project. It involves 23 different academic institutions across Europe, and all the major European pharmaceutical industries are represented as well. So my role in STEM Bank, obviously I have one specific role, it's a very large project, is to demonstrate and to optimize gene editing protocols that will be used by us and by our collaborators. And so in order to do this, I've used two genes essentially as our model systems. The first is LRRK2, mutations in which cause Parkinson's disease, which is a big interest in our lab. And the second gene that I'm looking at is the C9orf72. There's a repeat expansion recently discovered in this, which causes motor neuron disease. And so in today's talk, I'm mostly just going to talk in the first section about LRRK2 mutations that we've created, lines that we've created. And in the second, I'm going to look a little bit more at the application of iPS cell and gene editing technologies to look at a disease model. So LRRK2, it stands for leucine-rich repeat kinase 2. So this protein is a member of the ROCCO family of proteins. So they all contain this RAS complex proteins domain here and this C-terminal of ROCK domain here. In addition to that, LRRK2 also contains this kinase domain and these various putative lipid protein interacting domains. And as I mentioned, the reason that we're so interested in LRRK2 is that mutations in LRRK2 cause Parkinson's disease. And two of the most common mutations causing disease are this G2019S mutation here in the kinase domain and this R144.1C mutation in the ROCK domain. So another thing that's very interesting about LRRK2 is that nobody knows what it does. We really don't have a clue. It's a massive and complex protein. But we do know that it's not very highly expressed in neurons, but it is quite highly expressed in immune cells, and in T cells, B cells, monocytes, and in macrophages. And that's of interest to us because we have a protocol for generating, which we developed in our lab, for generating macrophages from iPS cells. So this is a very robust protocol. We simply generate embryoid bodies from our iPS cells. And then we grow them in MCSF and IL-3, and then by simply taking out the IL-3, leaving them in MCSF, the monocytes then convert to macrophages. And using this method, just with a single T175 flask, we're actually able to produce tens of millions of monocytes for our experiments. They will actually continue to produce for many months. So it's a very versatile system. So some of the genetic manipulations that are of interest to us and that we've carried out include just knocking out LRRK2, and the reason for this is to start to see if we can sort of probe for any phenotypes that might pop out. It's also a very useful immunoprecipitation control for our proteomics studies as well. The next gene edit that's of use to us and to our partners is demonstrating point mutation repair. In terms of LRRK2, we want to repair G2019S and R144.1C. These are to be used as isogenic controls in basic assays in RNA-seq as well. Finally, something else that we want to be able to do is tag our LRRK2 with a fluorescent protein, so we can do some live cellular imaging and look at cellular localization of the protein. Another use for this is for pull down for immunoprecipitation, because there's actually no good antibodies for LRRK2. So this is just an overview of a protocol which we sort of developed over a few years for editing iPS cells. So we begin by nucleoaffecting our iPS cells with our CRISPR-Cas9 constructs or donor construct, whatever it might be. And then we plate them down onto M-teaser and onto matrigel in M-teaser medium. And then we carry out a very transient puromycin selection. And this is because the plasmid, we've been mostly using plasmids for our gene editing work. And the plasmid that we've used is it's PX462, I think it is, from the Zhang lab. It's an all-in-one, and it contains a puromycin expression cassette. We then take these cells, the surviving cells, which we assume are highly transfected, are highly transfected cells, and then we passage them out at low density onto mouse embryonic fibroblasts, onto feeder cells. We then pick clones from there. It's a really laborious process, and plate them out onto 96 well plates of matrigel and M-teaser. We can then passage these plates, essentially creating clones of these plates. We create many, many clones. Some of them of these plates we freeze down. Others then we do genomic DNA extraction, and then we can carry out whatever analysis method it might be, identify the clones we're looking for, and then go back and thaw them and begin to expand them again. So to generate a knockout, it's a fairly, I think we've covered this in great detail today. It's a fairly simple procedure. All we do is we target a fairly early exon in the gene, and hopefully we'll generate a biallelic frame shift that'll knock the gene out of frame. And we screen all of these clones that we produce using high-resolution melt analysis. It's a really easy way of looking for knockouts. It's a system that we introduced a couple of years ago. All it really involves is determining the melting temperature of PCR amplicons. In that way, you can see anything that varies from a normal wild-type clone, which happens to be in green here. We then send out for sequencing and analysis. And here's one such clone that we've found, a 16 base pair deletion, a 37 base pair insertion, and so it's a biallelic out-of-frame shift. Looking down here, we see a Western blot. I should mention LRRK2 is upregulated by interferon. That's one of the few things we know about it. And we can see in the knockout there's no sign, at least there's no sign of a full-length protein, is what I should really say. One of the original stipulations in the STEM Bank project was that all gene repairs that we carry out should be footprintless. So this really means that there should be no other genomic change other than the particular mutation that you're trying to repair. So this precludes the use of selectable markers because you're going to leave behind LOXP sites. It also irritatingly means we can't silently mutate PAM sites and so forth, which means that you're going to get recleaving of already targeted or repaired alleles. So I had a hunch this wouldn't be very efficient. So to determine exactly how efficient it is, I use this rather nifty gene repair reporter system that we developed in our lab. I see that another group recently published it. Without checking, we had published it a year before them, but apparently it's all news. So all it consists of is simply a blue fluorescent protein expression cassette inside a lentivirus, which we then transduce into cells and stably integrate. By just changing, or let's say repairing 3 nucleotides in BFP, you can convert it to GFP, and then you can simply flow it and see exactly how efficient your gene repair has been. Of course, by then knocking out, if you don't see any blue fluorescent protein expression, that's signs of non-homologous end joining events as well. So it gives a nice readout. So to investigate footprintless gene repair, I transfected these cells with various donor constructs. First, a wild-type plasmid. This is essentially my footprintless approach. There's no mutation in the PAM. A mutated plasmid in which we have, a mutated plasmid in which we have mutated the PAM sequence, and wild-type oligo, and I've attempted to repair in the presence of either a double-stranded break or a single-stranded break. These are the constructs down here, and you can see the mutant donor has a mutated PAM, and the various nucleotides, or the 3 nucleotides required to convert it to GFP. And as you can see straight away, mutating a PAM massively increases gene editing efficiency 10-15-fold. So essentially, footprintless gene repair is extremely inefficient, and in iPS cells means we have to screen lots and lots of clones. So that's what we tried doing. So that's what we tried doing with, in order to repair G2019S, the G2019S mutation in LRRK2. So again, we're screening our clones by high-resolution melt analysis. It's so sensitive a technique, if you've got it set up right, that you can actually detect a single base pair change. So here are some patient lines that we've run through it. On the right here, you can see 2 wild-type alleles, and shifted here to the left, you can see 2 patient lines that contain G2019S mutation. And so we tried nicking our DNA and providing a double-stranded DNA donor, looked at several hundred clones, saw absolutely nothing. Tried with a double-stranded break and a double-stranded DNA donor, again, nothing. And we did actually get 2 clones back from 300 clones using a double-stranded break with a single-stranded oligodeoxynucleotide. So really, it's not a robust method. It's not easy to do. So and this is in comparison to footprintless gene repair, or non-footprintless gene repair, which we've essentially implemented in the project. So here is a typical donor for G2019S repair. And in it, I've mutated the PAM sites here and here, and I've also introduced a silent PST1 restriction site. But interestingly, you'll see another issue that pops up. And so here are some of these digest products obviously indicate that a recombination event has occurred. So we analyzed 258 clones, and of those clones, 17 contained recombination events. They produced digest products. But of those 17 clones, only 4 were correctly repaired. All the remaining basically had indels. So for 2 of them, the mutation was corrected, but the second allele was deleted. For 4 of them, we could tell that the wrong allele had been targeted, the second part of the second allele deleted. And then for 5, the correction couldn't be confirmed due to the nature of the indel. So basically, yes, so you can see the problem that can occur when your CRISPR-Cas9 or when your guide RNAs are able to target both alleles. However, in the case of 144.1c repair, the guide RNA luckily happens to run across the R144.1c mutation site. So in that way, by obviously designing the guide RNA only to recognize that site, it only targets 1 allele. And that shows in the repair efficiency that we get here. Out of 156 clones analyzed, we get 10 recombination events, and of those, 8 are correctly repaired clones. And we do see mutation corrected with second allele deleted for 1, and this is probably because of some slight off-target effects. And for the other, the wrong allele has been targeted, second allele has been deleted. Conversely, if you try to knock in R144.1c, it goes back to the same problem as before. In this case, where our guide RNAs are able to target either allele, which are both wild type, and of 248 clones analyzed, you get 5 recombination events, and only 1 is a correctly repaired clone. Yeah. Here's a nice little phenotype that we were able to identify in our G2019S repaired cells. So if you inhibit the kinase domain in LRRK2, what happens is you get dephosphorylation of serine residues in the protein, which leads to ubiquitination and protein turnover. What is interesting about G2019S, it actually causes hyperactivity of the kinase domain, which overcomes the effects of the kinase inhibitors. And in that way, you get less ubiquitination, less turnover of protein. You can see a reversion of that, a reversal of that phenotype in our repaired clones. So as I mentioned, another edit that we wanted to carry out was being able to fuse a fluorescent protein to the either CRN terminal of LRRK2. This is the construct that we used. The protein of choice was M-cherry, but hearing an earlier talk there, I'm starting to have doubts about that. This is the nature of the construct that we used. It contains because of the size of the DNA that we're trying to integrate at this point, we've had to use a selection marker. EF1 alpha is one of the few promoters that highly expresses in iPS cells, and it's driving a puro-TK fusion gene, so puromycin for positive selection, thymidine kinase for negative selection. This is all flanked by these LOXP sites, so it'll return LRRK2 back to its own control of its own poly A signal. And afterwards, we simply cree it out. These are the guide RNA locations. They're a bit of a distance from the stop site, and that does show in the efficiency. So 232 clones selected. So we simply analyzed by PCR of 200 with one of the forward primer here outside the homology arm. So of 232 clones that we analyzed, 6 contain the selection cassette at the correct location, so it's 2.6%. The rest are simply randomly integrated. And as you can see, here's a PCR of the polyclonal population. So we then nucleoaffected our cells with Cree messenger RNA, applied ganciclovir treatment to select for those that have lost the selection cassette. And we find this is actually a PCR of the polyclonal population, so it's been so efficient we can't even see the larger size band pop out. So we differentiated these cells to macrophages using the model I've described. This is a Western blot. In it, we can see using an M-cherry antibody, a single band pop up. Using the LRRK2 antibody, we can see that there's 2 bands have popped up, so it indicates that this is a single allele that has been targeted. This is just some confocal microscopy images of, from, as I said, that previous talk. This is obviously a lysosome-associated M-cherry, but okay. So I'm going to change gears here a little bit. I'm going to talk about some of the more recent work that I've done with motor neuron disease, which is also known as amyotrophic lateral sclerosis. Oops. So motor neuron disease, it's characterized by motor neuron degeneration and progressive paralysis, which ultimately results in respiratory failure and death. We know that about 95% of the cases are sporadic, and the remaining 5% being familial. And of those 5% familial cases, about 25% are explained by mutations in these genes, superoxide dismutase and so forth, which leaves the question, what is causing the other 75% of cases? Linkage analysis studies just a couple of years ago identified this repeat or expansion here occurring on chromosome 9, which seems to explain about another 50% of cases. It's inside in this open reading frame here, which does encode for a protein of unknown function. And depending on the transcript variant that you're looking at, it's either in the first intron or it's in the promoter region, the repeat expansion. What we find is that in healthy individuals, there's about less than 20 repeats of this hexanucleotide sequence. ALS patients, on the other hand, have a minimum 30 repeats, and it can go up to thousands of repeats. It's a massive, massive sequence. So the next question is, well, how does this repeat expansion cause disease? And there are three hypotheses based on various observations that may explain this. Either one on its own, or more likely to be several of these involved, of these factors. So the first is haploinsufficiency. We do see reduced transcript levels in patient cells. Also, that is backed up by some animal model evidence. In C. elegans, if you generate null mutations in the C9orf72 ortholog, you do see some neuronal degeneration effects. And I think in the zebrafish model as well, with siRNA knockdown, you do see axonal shortening and so forth. The second potential mechanism of disease is RNA toxicity. These big repeat transcript sequences are actually transcribed, and they appear to aggregate in the nucleus. And one theory as to what's going on there is that there may be sequestering RNA binding proteins that are involved in splicing. The third potential cause of disease is we do get what's known as a RAN translation of these repeat sequences. So RAN translation is a repeat-associated non-ATG translation, and so you get these dipeptide proteins popping out, and these do clearly aggregate in the cell as well. So the stimulus for this work was this paper that just came out earlier this year, in which the authors generated C9orf72 homozygous and hemizygote knockouts, mouse knockouts. And instead of sort of going down the more classical route of focusing purely on the central nervous system, the authors actually cast a slightly wider net and discovered enlarged lymph nodes and spleens in homozygote knockout mice, not in the hemizygote knockouts, I should say. Doing a little bit of histological analysis, and what they found was these large debris-filled cells, which were CD11b positive staining, which was likely to be macrophages. To test this theory a little further, they took some monocytes from the blood of these mouse and generated macrophages. And just a little snapshot of some of the results they found. There was no difference in phagocytosis and uptake of zymosin particles amongst the different blood monocyte-derived macrophages. However, after zymosin particle uptake, zymosin is sort of a yeast particle. After uptake, there was this sudden increase in reactive oxygen species production in both the homozygote and the hemizygote knockouts versus the wild-type control. Interestingly, and in response to various stimuli, LPS, PAM, PGN, and CPG, they also saw increased TNF-alpha secretion in both the homozygote and hemizygote knockouts versus the wild-type controls. So, well, this definitely suggests that haploinsufficiency causes some sort of pro-inflammatory phenotype in macrophages. They've also actually repeated these experiments in microglia that they isolated from spinal cord and got the very same results. So the authors sort of came up with this dual mechanism model, perhaps where we're seeing gain of function mutations in neurons, but maybe loss of function in microglia, and maybe the two come together to produce the neurodegeneration that we're seeing. But of course, this is all in mouse. It's not in humans. We can't create isogenic humans as of yet. And so another little niggling thing about this is that they're also the haploinsufficiency model is a knockout. It's not introducing massive repeat expansion, so it's not really very close to the actual disease model. So one way we can look at this in greater detail is using iPS cells. So what we looked to do in patient cells is we wanted to revert this massive this massive repeat expansion from 1,000 repeats back down to just 2 repeats of sequence. And this proved easier to do than we had actually anticipated. It sounds like a difficult project, but it's actually it went quite smoothly. So we simply picked some guide RNA sites quite close to the repeat expansion. We used a donor with just 2 repeats, and we also used this. We also had to use this same selection cassette as I described earlier on, with EF1 alpha driving pure OTK. We analyzed clones by PCR, which you can see here, and we found several that had the 3.4 kb product that we were looking for. We also ensured we used digital droplet PCR to ensure that we were only one copy had actually integrated. We also used repeat primed PCR. This is a method of amplifying repeat expansion or repeat expansions. You can't simply use standard PCR to amplify 6,000 nucleotides of gene C. It really won't happen. We were able to show, here's a wild type line. There's 6 repeats. In our gene edited line, there's only 2 repeats. And here's the line before we carried out our gene editing, where we can see 50 plus repeats. I should say that the maximum number of repeats that you can see with repeat primed PCR is about 50. So we did a little characterization of these clones that we produced. They're still pluripotent. We can see nanog, OCT4, and Tra160 staining. We also carry out, because of the stem bank project, pretty comprehensive karyotype analysis using cytosnip as well. And as you can see, these retain all the characteristics of the parental line. We then used that protocol that I mentioned earlier to differentiate these iPS cells into macrophages. We characterize these macrophages. As you can see, as macrophages should, they take up zymogen particles. We also stained them for various markers, CD11b, CD14, and CD45, to show that these were monocytic cells. Finally, we also looked at C9 or 72 RNA expression levels, just to make sure that they were as expected, and they did actually produce the same sort of levels of transcripts as in blood-derived or blood monocyte-derived macrophages. So then we carried out some assays to see if we can also see what has been seen in the mouse model in a human relevant model. In phagocytosis, as before, we saw no difference. They all take up zymogen particles to the same level. Reactive oxygen species, again, we don't actually see a difference, even though they did report it in the mouse model. However, in response to various stimuli, the patient lines did produce more TNF alpha versus either our genetically edited control or the standard wild type control. So that result is the same as is seen in the mouse model. We also carried out some a cytokine protein array just to see if there was any other upregulation. And even at basal level, we see increased IL-10, RANTES, IL-17E, ICAM, and interferon gamma in the patient lines versus these genetically modified patient controls. So just to demonstrate how versatile the iPS cell model is, our collaborators were able to take the same lines and differentiate them out into motor neurons using a protocol they've developed. They'd stain them with various markers, HB9, SMI32, and so forth, just to confirm that these are actually motor neurons. And they made some very interesting observations. So they were actually able to detect in their patient line motor neurons. They can actually see, it's a little bit difficult to see, but you can actually see little RNA foci popping up that have been removed, or that are absent in the genetically edited controls. In addition to that, they also looked at C9orf72 transcripts, and they can also see this reduction in transcript levels in the patient lines versus either the genetically modified or the wild type controls. Also, these repeat-associated dipeptides, again, higher levels in the patient of all the dipeptides that occur versus the genetically edited isogenic control or standard control. Again, a couple of other observations. Enhanced cellular stress, as measured by poly-A binding protein, was observed in the in the patient lines versus either the isogenically edited lines or the wild-type controls. And again, enhanced apoptosis was also observed. So just to summarize these results, so I've described our gene editing protocols in iPS cells. I've compared footprintless versus non-footprintless gene editing. You can see exactly how inefficient the footprintless approach is. I've described some of the LRRK2 gene edits, knockouts, repair, knock-in, and gene tagging. I've looked at how we've managed to shorten a massive hexanucleotide repeat expansion back to wild type size through gene editing, and I've shown some of the results of the phenotype analysis. And this is a more, as I say, this is a human relevant cell model, which is showing the same results that has been produced in a mouse model. With that, that's the end of my talk. I just want to acknowledge everybody involved in the James Lab, my supervisor Sally Cowley, our lab head William James, and our collaborator on the C9orf72 project, Kevin Talbot and Nida. And obviously I'd like to thank STEM Bank for all the funding too. Thank you very much.